For years, peptide therapeutics have held incredible promise in oncology, precision tools with minimal off-target effects. Yet time and again, they've stumbled on the same hurdles. How do you get them inside a cancer cell, and how do you keep them intact long enough to actually work? It's been a frustrating bottleneck for true innovation. But what if we could chemically engineer a solution? A new method just surfaced, and for those tracking the cutting edge, it's a game changer, potentially unlocking a new era for cancer treatment.
The core problem stems from the inherent fragility of linear peptides, easily degraded and notoriously poor at penetrating cell membranes. This novel bis-urea stapling technique directly confronts these issues. It covalently links specific amino acid residues, creating a rigid macrocyclic structure. What's truly brilliant is its chemoselectivity. It forms these bis-urea bridges precisely between native amino groups, either lysine-lysine or an N-terminus lysine pairing, crucially preserving sensitive residues vital for biological activity. Think of it, arginine, histidine, tryptophan, all untouched. Mess with those and your drug is inert. The immediate benefits are substantial. We're seeing improved membrane permeability, non-negotiable for intracellular targets, and significantly increased stability against both chemical and proteolytic degradation. A peptide that can survive the harsh environment of the human body to actually reach its target. That means longer half-lives, potentially less frequent dosing, and ultimately a more favorable pharmacokinetic profile. Clinically, that translates directly to better efficacy and patient compliance.
This isn't just elegant chemistry, it's about tangible clinical impact. Initial in vitro and in vivo data for oncology applications are highly encouraging. These stapled peptides demonstrate potent cytotoxicity in diverse cancer cell lines, inducing dose-dependent apoptotic responses in tumor models. This isn't just killing cells, it's programmed cell death, a cleaner, more effective way to eliminate cancer without excessive collateral damage. It's evidence-based optimism. If you find this deep dive into drug development as compelling as I do, hit that subscribe button and tell us, what breakthrough peptide research are you tracking right now?
Now, the mechanism of action here is key. Many of these stapled peptides disrupt critical protein-protein interactions vital for cancer cell survival, like the p53 MDM2 pathway. By being rigidified into their bioactive conformations, they bind more effectively to intracellular targets typically inaccessible to larger antibodies or less specific small molecules. But let's be the regulatory realist. The path from lab to patient is arduous. For a novel technology like bis-urea peptide stapling, we're firmly in discovery and lead optimization. Before an IND application, extensive GLP toxicology assessments are mandatory: acute, subacute, chronic toxicity across species, detailed ADME profiling. We need to evaluate the metabolic fate of that bis-urea bridge itself. Are there toxic degradation products? How does it affect immunogenicity? Realistically, we're looking at several years before human clinical trials commence. Optimistically, 2-4 years for preclinical, then 6-12 months for IND filing, followed by 1-2 years for phase 1. Scalability for GMP-compliant manufacturing is another critical factor. Can we make enough, consistently, without breaking the bank?
The outlook for chemoselective bis-urea peptide stapling is genuinely exciting. By addressing fundamental stability and permeability issues, this technology could transform intrinsically active peptides into truly viable therapeutic agents, particularly for intracellular oncology targets. The ability to create stable, cell-penetrating peptides that induce apoptosis with dose-dependent precision offers a significant advantage. It has the potential to establish a new paradigm for peptide drug design, not just in oncology, but for a range of diseases requiring intracellular modulation. This technology could undrug previously inaccessible protein-protein interactions. It's a realistic assessment. This method significantly expands the therapeutic utility of peptides, positioning them as key players in future pharmacological interventions. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment. Stay ahead of the clinical curve. The next great peptide is already in phase 2. I'm Sonia Rao, and you've been listening to Peptides. Today.